Welcome, curious minds, to another deep dive. Today, we are plunging into a story that, it begins in the hushed, dust-filled chambers of ancient Egypt, and somehow, remarkably, pivots to the absolute cutting edge of modern medicine. I mean, imagine this. Centuries of whispers, right? Sealed tombs, the chilling legend of a pharaoh's wrath, striking down anyone who dares disturb the dead.
It's classic stuff, straight out of the movies.
Exactly. The infamous curse of the pharaohs. But here's the real paradox, the genuinely mind-bending twist we're getting into today. Something once feared, like really feared as a symbol of death, it might now hold a critical key to life.
The potential cure, even, for one of our deadliest diseases.
It's wild, and it perfectly encapsulates how science can just completely transform our understanding, doesn't it? Moving from myth to medicine.
Absolutely. From the eerie silence of a burial chamber to the buzz of a modern lab. And at the heart of it all, a fungus. Aspergillus flavus.
That's the one. And for this deep dive, we're digging into a really fascinating report. It came out in News 18, June 25, 2025. So our mission today is pretty clear. Pull out the key nuggets from this amazing intersection of history, myth, and, well, biology. We're going to trace the path of this so-called curse and see how it's being completely reevaluated, not just as an explanation for old deaths, but as a potential cure in the fight against cancer. Okay, let's unpack this layer by layer.
It's a fantastic illustration of how science can peel back these layers of mystery, reveal truths you just wouldn't expect, often hidden in plain sight or maybe things we were afraid of.
Totally. And where else can we possibly start but with the legend itself? I mean, the curse of the pharaohs, it's had this incredible grip on our imagination for, what, a century now?
At least it's deeply rooted in ancient Egyptian lore, this idea, this warning.
Yeah.
Doom for anyone who violates a pharaoh's tomb or, disturbs the mummy.
The idea being that these powerful rulers, even dead, still have some kind of protective power, a vengeful essence guarding their stuff.
That's the core concept, yeah. And look, curses to protect tombs weren't unique to Egypt. Lots of ancient cultures had similar warnings. But the pharaoh's curse, it just exploded into global consciousness.
Right. It became this huge pop culture thing.
Largely because of one very specific, very dramatic event.
King Tutankhamen's tomb, 1920s. Howard Carter, Lord Carnarvon.
Exactly, November 1922. That discovery wasn't just archaeology, it was a global media sensation. Front page news everywhere. People were obsessed.
Thrilling stuff, untouched treasures, ancient secrets revealed. The world was watching as they broke that seal.
But then things started to happen. What followed really fueled that eerie tale of the pharaoh's wrath. It turned this scientific triumph into, well, a kind of macabre legend.
Because people connected to the digs started dying or getting seriously ill.
Right. A chilling chain of events, almost too perfectly timed for the myth makers.
Who are we talking about specifically?
Well, the list became almost as famous as the treasures. Lord Carnarvon himself, the expedition's financier, he was one of the first, died just months after the opening, April 1923. Officially, it was complications from an infected mosquito bite, erysipelas, then pneumonia.
But the timing.
Yeah.
Wow. Just months after going in.
Unnervingly precise, wasn't it? Then there was George J. Gould, an American financier, friend of Carnarvon, visited the tomb shortly after, fell ill with pneumonia, died May 1923.
Another one.
And Arthur Mace, an Egyptologist, he was part of Carter's core team, actually helped break into the burial chamber. He died in 1928, mysterious illness, lung problems. These weren't unknown people.
No, prominent figures.
Yeah.
And dying one after another right after opening this incredibly sacred place. You can see why the story took hold.
Oh, absolutely. The media went wild with it. It created this frenzy, and you can understand why, can't you? It plays on primal fears.
The unknown ancient power messing with sacred things.
Exactly. These stories are potent, they grab our imagination, and crucially, even when scientific explanations start to trickle out, these legends stick around. They shape how people see things.
People love a good mystery, right? Especially one with ancient curses and gold.
Definitely. But what's really fascinating here is how science eventually, methodically, started to demystify it without, ruining the intrigue.
Okay, so how did science explain it? What was really going on?
Later investigations, using modern microbiology, environmental science, they offered a much more, earthly explanation. The idea wasn't a vengeful spirit. It was something biological, something lurking in those sealed chambers for millennia.
Dormant, waiting.
Exactly, waiting for the seal to be broken.
So not a supernatural curse, but a biological one.
Pretty much a toxic fungus, not as dramatic maybe, but definitely dangerous. silent.
Which fungus was it?
The key culprit identified was Aspergillus flavus. Now, this isn't some exotic creature. It's a really common mold found in soil, decaying stuff all over the world.
So why was it so dangerous in a tomb?
Because in that unique environment, sealed, undisturbed for thousands of years, probably humid, nutrient-rich from, organic materials in the tomb, this particular fungus can produce potent mycotoxins.
Mycotoxins, like poisons.
Exactly. Specifically, a group called aflatoxins. These are some of the most potent, naturally occurring carcinogens we know of. They can also suppress the immune system.
Wait, carcinogens, so cancer-causing toxins.
Uh,
From a mold.
Absolutely. That's a crucial detail. So picture this. The tomb is breached, air rushes in, disturbs these ancient, hardy spores. They've been sitting there dormant for centuries.
And suddenly they're airborne.
Airborne, carrying these toxins, and the people entering the tomb inhale them, deeply probably given the dust and effort.
Oh, wow.
For anyone with maybe a pre-existing condition or even just a bad reaction to a high dose of these spores and toxins, this exposure could cause severe respiratory problems, weaken their immune system, or, contribute directly to their deaths, especially back then, without modern treatments.
That makes a scary amount of sense. It's almost more chilling because it's real. A microscopic killer, preserved by time.
It completely shifts the perspective, doesn't it? From the mystical to the microbial. And it's arguably more insidious because it's tangible, scientifically plausible.
So it wasn't just Tutankhamun's tomb, presumably. This understanding changed how archaeologists approach these sites.
It absolutely did. It wasn't just about ghosts anymore. It was about real biohazards. Modern archaeology takes precautions, respirators, air quality monitoring, partly because of this revised understanding, moving from superstition to, actual risk assessment.
Okay, so science explains the curse. End of story.
Not even close. Because here's where it takes this incredible, almost unbelievable turn. This exact fungus, Aspergillus flavus, the one linked to death and the curse, it's reemerged in a totally new light.
How so?
It's gone from being the villain of the story, the microscopic menace, to potentially holding a key to modern medicine, from historical danger to perhaps future hope.
You're kidding. The same fungus.
The very same. It's a huge paradigm shift, a 100-year leap, really, from that dusty tomb filled with fear to a sterile, high-tech lab filled with promise.
Okay, my mind is officially blown. A century after King Tut's tomb kicked off the curse frenzy, what's the news?
Researchers at the University of Pennsylvania, a groundbreaking study, published very recently, and it strongly suggests Aspergillus flavus might hold the key to fighting cancer.
Cancer. The tomb fungus fights cancer. This is where it gets really interesting.
It really does. The research came out in a top journal, Nature Chemical Biology. That tells you it's serious, rigorous science. What they found is that this ancient fungus, it doesn't just make toxins, it also produces a specific class of molecules that fight cancer cells.
Wow, have they named them?
They have. They're calling them asparagmecins. It's just a fantastic example of how nature keeps surprising us, revealing its hidden chemical weapons in the most unlikely places.
And there was that quote you mentioned from Sherry Gao at Penn.
Yes, Professor Sherry Gao, Chemical and Biomolecular Engineering. It really sums it up perfectly. She said, "Nature has given us this incredible pharmacy. It's up to us to uncover its secrets."
Nature has given us this incredible pharmacy. That's powerful. It reframes the whole natural world, doesn't it? Not just stuff to study, but a resource for healing.
Absolutely. And look, this isn't a totally new idea in principle. Think about penicillin.
Right, from the penicillium mold, revolutionized medicine.
Exactly.
Huh?
Discovered by Fleming. Fungi giving us lifesavers. This discovery with Aspergillus flavus echoes that history. It underlines the power of what we call bioprospecting.
Bioprospecting. Okay, break that down for us. What does that actually involve? Is it just randomly looking for stuff in nature?
Not random at all, ideally. It's systematic. It brings together scientists from different fields, ecologists, microbiologists, chemists, geneticists. They meticulously search diverse ecosystems.
Like rainforests, oceans.
Exactly. Or deserts, deep-sea vents, places where organisms have had to evolve unique ways to survive, compete, defend themselves. They develop complex chemical compounds over millions of years.
And those defense chemicals can sometimes help us.
Often, yes. They can have amazing therapeutic potential. Think about it. Nature's been running R&D for billions of years, developing these incredibly potent, refined molecules. We're just now getting good at finding them and understanding them, even in places like, well, ancient tombs.
So it's like tapping into evolution's chemical library.
Precisely.
Yeah.
It reminds us that even with all our fancy synthetic chemistry, nature might still hold the best drugs. We just need to know where and how to look. It's about decoding nature's secrets.
Okay, let's get into the specifics then. How is this fungus, Aspergillus flavus, fighting cancer? What did the Penn team actually do?
Right, the methodology. They started systematically. They studied about a dozen different strains of Aspergillus, not just flavus, but related species too, collected from various places.
And screen them against cancer cells.
Yes. They isolated compounds from these fungal strains and tested them against leukemia cells in the lab. And the initial results were really promising.
How promising.
Some of these compounds showed strong anti-leukemia activity even without any chemical modification, just as they were produced by the fungus.
That's significant, right? That they worked straight out of the box, so to speak.
Hugely significant. It highlights the inherent potency. It's rare and incredibly exciting when you find a natural compound that's already active against something like leukemia cells without needing chemists to tweak it first.
And leukemia is a tough one. Were they targeting specific types?
Yes, the report mentioned activity against several human leukemia cell lines, including models for acute myeloid leukemia, AML, which is notoriously difficult to treat. So the relevance was immediate. But of course, they didn't stop there.
They wanted to see if they could make it even better.
Exactly. Can we boost this natural power?
Yeah.
And this is where that really unexpected twist comes in.
The royal jelly thing, this still sounds bizarre to me.
It is bizarre, but that's science sometimes, right? Serendipity trying unusual combinations. They found that by adding a compound derived from royal jelly.
The stuff bees feed their queen.
That exact stuff. Adding it to the asparagmasins significantly enhanced their potency against the cancer cells. It's this amazing cross-kingdom synergy. Fungus meets bee product.
That is just wild. So what happened to the effectiveness when they added the royal jelly component? How good did it get?
The results were genuinely impressive, clinically relevant even at this early stage. The enhanced asparagmasins showed effectiveness that matched established chemotherapy drugs.
Matched, like standard chemo drugs used right now.
Yes. The report specifically mentions cytarabine and daunorubicin. These are frontline treatments for acute leukemia. To have a new compound, derived from a fungus in royal jelly, match the power of these established drugs in lab tests. That's a huge deal.
Okay, that provides real context. It's not just it works a bit. It works as well as the current standard of care, potentially.
In these specific lab models, yes. It elevates it from just an interesting finding to a genuinely promising therapeutic lead, something that could, down the line, make a real difference.
But is matching enough? Does it offer any potential advantages over those existing drugs? Maybe fewer side effects or working against resistant cancers? Or is it too early to tell?
That's the million-dollar question, isn't it? And yes, it's early days. These are in vitro results, lab dishes, not patients yet. But the potential advantages are exactly what researchers will be looking for. Could it have a different mechanism that bypasses resistance? Could it be less toxic to healthy cells?
Because current chemo can be pretty brutal.
It can be, yes, because it often targets all rapidly dividing cells, not just cancerous ones. If these natural compounds have a more specific action or a better safety profile, that would be a massive win. Matching effectiveness is the first hurdle. Potential advantages are the next goal, and that requires a lot more research animal studies than human trials. But the potential is definitely there.
Okay, so how do they work? On a cellular level, what are these asparagmasins actually doing to the cancer cells?
Sherry Gao explained it. The mechanism involves microtubules.
Microtubules. Okay, refresh my memory. What are those?
Think of them as the cell's internal scaffolding, or like tiny highways. They're crucial for maintaining cell shape, moving things around inside the cell, and critically, they're essential for cell division.
How so?
When a cell divides, microtubules form this structure called the spindle. It's like a set of ropes that pull the copied chromosomes apart, ensuring each new daughter cell gets a complete set. It has to happen perfectly.
And cancer is all about uncontrolled division.
Exactly. Rapid, chaotic division. So these asparagmasins work by blocking the formation of microtubules. They essentially gum up the works, preventing the spindle from forming properly.
So the cancer cells can't divide.
Precisely. They get stuck in the division process and often die. It's a well-established anti-cancer strategy. Several existing chemo drugs target microtubules. Taxol is a famous example.
So it's a known vulnerability in cancer cells.
Right.
But this is a new compound exploiting that vulnerability, a new key for that lock.
That's a great way to put it. A new molecular tool targeting a proven weak point. And the research went even deeper. They found a specific gene involved in getting the drug into the cell.
Ah, right. The transport mechanism. Tell me about that.
They identified a gene called SLC46A3. It seems to act like a doorway, helping to transport the asparagmycins across the cell membrane and into the cancer cell.
Why is knowing that doorway so important?
It's huge for drug development. If you know how the drug gets in, you can potentially optimize it, maybe design variations that use the doorway more efficiently, or understand why some cells might become resistant. Perhaps they shut down that doorway.
So you could target delivery better, make it more potent at lower doses, maybe fewer side effects.
Exactly. Or overcome resistance. Understanding the transporter is key for rational drug design. It moves beyond just this works to this is how it works and here's how we might improve it. That level of detail is crucial for turning a promising compound into an actual medicine.
Finding the secret entrance and then figuring out how to, upgrade the delivery truck. That's really fascinating.
That's a good analogy, and that deep understanding is what paves the way for safer, more effective therapies.
Okay, this is obviously huge for Aspergillus flavus. But surely the implications go wider. If this fungus has these capabilities, what about all the other fungi out there?
That's exactly where the research points, and it's perhaps the most exciting aspect. The team didn't just find the genes for making asparagmycins in Aspergillus flavus. They found similar clusters of genes, biosynthetic gene clusters, in other fungal species, too.
Similar gene blueprints in other fungi, meaning
Meaning this might not be a one-off. The ability to produce these kinds of potentially anti-cancer compounds might be more widespread in the fungal kingdom than we thought.
so it's not just one magic fungus. It could be a whole class of fungi making similar, maybe even better compounds.
Potentially, yes. It opens up this massive unexplored territory. Kiyu Ni, another researcher on the project, had a great quote about this. She said, "Even though only a few have been found, almost all of them have strong bioactivity. This is an unexplored region with tremendous potential."
Strong bioactivity.
Yeah.
Unexplored region. That sounds incredibly optimistic, like we've just scratched the surface.
That's the feeling, yes. Strong bioactivity means these compounds pack a punch even in early tests. They have a real, measurable effect. And the fungal kingdom is vast, millions of species, most barely studied. Each one is a potential source of novel chemistry.
But aren't natural products notoriously difficult to work with? Getting enough material, synthesizing complex molecules?
Historically, yes, those have been major challenges. Isolation, purification, scaling up. Getting enough of the compound from the original fungus can be tough. And chemically synthesizing these intricate molecules is often incredibly complex and expensive.
So how do we overcome that to tap into this potential?
Technology is catching up. Advances in genomics, synthetic biology, genetic engineering, they're game changers. Now, researchers can identify the specific genes the fungus uses to make the compound.
The biosynthetic gene cluster you mentioned.
Exactly. Once you have that genetic blueprint, you can potentially transfer those genes into something easy to grow in the lab, like yeast or bacteria. Turn them into little factories churning out the compound.
So you don't need tons of the original rare fungus, you just need its recipe.
Precisely. You harness its genetic instructions. This makes large-scale production much more feasible. It bridges the gap between discovering something amazing in nature and actually being able to produce it as a drug. It allows us to really leverage nature's chemical genius.
Which brings us back to you listening to this. How does this whole story change your perspective on something like, mold, or even on ancient legends?
It's a profound shift, isn't it?
For centuries, this fungus was either invisible or feared, maybe blamed for a curse. Now, it could be a lifesaver.
What was literally feared as a symbol of death might become a beacon of hope. It really challenges us to question our assumptions, doesn't it? To look beyond the surface.
How many other things in nature do we fear or dismiss that might hold hidden benefits if we just look closer with the right tools?
It makes you wonder, how much potential is locked away in organisms we haven't studied or even discovered? It urges that critical thinking, looking for multiple angles, reconsidering what we think we know.
It's just an incredible narrative arc, from the Pharaoh's curse to a potential cancer cure, Aspergillus flavus. It's not just a footnote in archaeology anymore.
No, it could make serious medical history, a legacy far more impactful than any tomb legend. It's a potent reminder that our understanding of the natural world is constantly evolving, often in really startling ways.
The interplay is just amazing. History, myth, cutting-edge science, all converging on this one fungus.
It really shows the value of looking at old mysteries with new eyes, new technology.
Absolutely. So what an incredible deep dive today. From Tutankhamun's tomb and those eerie curse stories, all the way to a modern lab where that same fungus shows promise against cancer. We've unpacked the superstition, the science behind the curse, and now this amazing medical potential.
It highlights the power of questioning, of digging deeper, of not accepting the first easy explanation.
It really does. And maybe a final thought for you to take away. As you reflect on this, think about how many other curses or feared bits of nature throughout history might just be waiting for science to uncover their hidden cures.
Or other surprising benefits we haven't even imagined yet.
Right. Consider that delicate balance in nature. The same organism, a threat in one context, a potential savior in another. It all depends on how we interact with it, how deeply we understand its biology, its chemistry.
It's a powerful case for interdisciplinary work, isn't it? Connecting archaeology, microbiology, chemistry, medicine. And it's a reminder that nature still holds so many secrets, vast potential, just waiting.
Waiting for us to find it. A truly powerful idea. Until next time, keep exploring, keep questioning, and keep diving deep.